He analyzed them chemically, functionally. He did everything. Chemically, it means he even broke them down to understand their composition. They are composed of 80% water. They are composed of carbon, hydrogen, oxygen, nitrogen, like all living organisms. They are composed of minerals. There you go. And he noticed differences depending on their, the microzymas of almonds, for example. They have much more minerals than the microzymas from other vital centers where he took samples. He noticed some differences, but overall, we always find the same elements found in all living organisms. These are the vital elements. They perform specific functions. They produce enzymes. I've emphasized that quite a bit. These chemical analyses he did were in terms of fermentation, meaning each time. And that's important too. I'm coming back to that. I must have already said it, that fermentation depends on the environment. Depending on the fermentation, there's no alcoholic ferment, lactic ferment, or that. These are things that must, no? Does that make sense to you when I say that? There's no lactic ferment, no alcoholic ferment, no ferment, right? Depending on the environment, if we put it in sugar, we'll particularly get alcohol. But if we put it in starch, we'll get other products, particularly alcohol. And alcohol is a term for all fermentations. So at that point, all ferments are alcoholic. Actually, in our bodies, Bechamp analyzed, or his members of his team, they found alcohol everywhere. Well, not in the same quantity as when you find alcohol everywhere. Because all transformations that occur in the cell, in our organisms, all transformations are fermentations. But on the other hand, he found differences depending on the vital center. That is, take the liver or he takes the pancreas. First, the microzymas of the pancreas. He found them in a sheath of fat. By the way, he had to find them, because so he cleaned the seed thinking it was just lipid granules, probably. And then after cleaning, treating, well, he found something very small, even smaller than what he had found in the liver. And then he observed them, then he managed to make them multiply, ferment, produce enzymes. Each time he obtained something. And these granules, whether they come from the liver or the pancreas, do not act the same way. He tried, in experiments, he tried to take experiments on blood. Well, I don't really like experiments that are it's true that the poor animals, we make them undergo impossible things. And maybe that's why I realized that maybe that's why I didn't continue in biology and biochemistry, because I didn't want to work on animals, yes. I couldn't do it. I had a lot of trouble with that. And so, in particular, if he put the microzymas taken from the liver into a dog's blood, well, the dog survived in the same proportions. If he put the microzymas from the PR taken from the pancreas in the blood of a dog, the dog did not survive. So there was really a specificity. And well, he already noticed at the level of fermentations that he didn't always get exactly the same things, or there you go. There were variations in the fermentations. So he fermented on different types of environments. And so he had specificities unique to the vital center. He realized microzymas could build cells. These specificities unique to the vital center, he realized especially that, they were the same as those performed by the cells themselves. He had already studied the metabolism of cells before discovering microzymas, and he realized he was reproducing the same specificities with microzymas, the same fermentation products. And so he realized that microzymas performed the functions of the cell. He said to himself, but what is the cell in all this? Well, the cell, they build the cell to preserve the environment, to perform their function. Because the function is specific to a particular environment. Ultimately, the environment is suited to a specific function, and the microzyma needs a specific environment to properly perform its function. If we change the environment for a long time, or it will eventually adapt. Because these little creatures, they have incredible possibilities. It will eventually adapt. That is, and he realizes, for example, these pancreatic microzymas, if he made them digest fibrin. So if he put them in an environment with fibrin, well, the pancreatic microzymas later, if he injected them into a dog's blood, they no longer had their harmful aspect. So they had adapted. So they perform the functions of the cell. They perform the functions of the vital center, quite simply. They can adapt, so they belong to a vital center. They're able to adapt. But for them, in a living organism, they're meant to carry out the functions they acquired during embryonic development, and afterwards, they need the cells. Why the cells? What are the cells for? Well, to maintain this environment. They don't have any other role, ultimately. They have a role, perhaps, there you go, the DNA of the cells mainly has a utilitarian role for the microzymas. That is, they may have built a small machine. The cell is a small machine they built to be more efficient in their function. But it's the microzymas, they are the living. This is also true for plants. So they can build cells or develop into microorganisms. If the environment is unbalanced, it's all a matter of environment. And Antoine Bechamp drew what he observed. He didn't have techniques to photograph at that time. What they observed, he drew everything. He has, for example, an experiment on the sea of vinegar. There, from figure 1 to figure 4, what do you see? The development of bacteria. There, the long threads you see there. These are what we call leptotrix. They are very long bacteria. The small dots we have, this dark mass, all of it, all the small granulations there, these are the microzymas. And in the environment in which he had placed them, these microzymas transformed into bacteria. So from figure 1 to figure 4, all of this is the evolution of microzymas into bacteria. It was in the same environment that inevitably evolved, since over the course of fermentations there are eliminations. We must always remember that the environment keeps evolving. That's where I said the cell is important, because the microzymas of the cell will do the sorting. That is, gradually, they will continue to maintain this environment all the time, all the time, all the time. Meaning they will only let in what is beneficial for the cell, only let out what will help maintain the balance of the environment. These microzymas from the sea of vinegar in another environment, they manage to reconstitute cells and form cells. So there, in the mass, in the mass we see there, so it must be C. We see the granulations, and we see granulations starting to come together and form cells. And little by little, the cells detached. It's all a matter of environment. If the environment is balanced, we will have cells. The microzymas will perform their function and thus produce cells when cells are needed or something else. Everything in our organisms comes from them, since they were the only living elements in the yolk. And when it's unbalanced, they can evolve into bacteria. Evolve into bacteria in an environment conducive to bacterial evolution. If we put them in an environment conducive to mold evolution, they will evolve into molds. So it's the vital element of every living organism. They are unique to the organism and even at the vital center. And they are. I was saying that they are the foundation of every living organism, so they organize. It's important, the organism, because everything will find its place within an organism. They organize, they are the foundation of everything. And when the organism is dead, maybe due to disorganization or when at some point the environments are so unbalanced that we inevitably shift towards something that is no longer possible, leading to the death of the individual. Well, they are still there. They are still there. And at that point, they move into a stage of decay. They will produce decay, meaning they will produce another type of fermentation. Instead of working together for the organism, this time they will work alone, autonomously. And at the end of decay, they can go through the stage of bacteria, through different stages. They will go through all the stages depending on the environment as time goes on, and airborne germs can also intervene. They might not, as long as, why not, there are others who will feast on the meal there in passing, because the organism is dead. Previously, they did not enter our organisms, because Antoine Bechamp observed that. He observed, in fact, he supposed it, but from all his, he wasn't really sure, but from everything he had observed, the microzymas active in our bodies are our own, and foreign microzymas do not enter an organism into an organism because they are not at home. However, from the stage of death, they start to intervene, since after all they find themselves in front of an inert substance. And so our own microzymas, helped by others, we decay. And what do we find at the end? Once everything has been reduced to almost nothing, to dust, we find the living microzymas. At the end of every living organism, we could do this for a leaf, we could do this for anything. Everything returns to its original state. We have become so accustomed to seeing things that they seem normal to us. Why does meat rot? Why does a plant rot? We think it's just a reaction of chemical reactions No, it doesn't happen on its own. Normally, inert matter, that's what I mentioned at the very beginning. There is a provoking cause, and this provoking cause is the microzymas. They are the ones who feed on everything. Matter, they are the ones who transform it. If there weren't these ferments, a leaf would remain a leaf. It wouldn't degrade. We wouldn't degrade either. So everything we see decay is a living act by the microorganisms. Everything is reducible to the microzyma. Right from the very beginning, in the yolk, there were microzymas. In the seed, we had microzymas. Everything was built from these small vital ferments. So they are truly the vital ferments. And they are the ones at the base of decay, who are the basic principle of decay. And they themselves are small vital organisms. But they are not decayable, because they are the basic principle of decay. And so what do we find in the air? In the environment, we find the remains of living organisms, animals, and plants. And what we believe to be microbes born to attack us are actually the remains of living organisms. It's essential what I'm saying here. It's essential. That is to say, the vision of the microbe we had is something that attacks us, that aggresses us. In reality, it's exactly the opposite. What we have in the air, the germs we find in the air, are the remains of living organisms, animals, and plants. It changes everything. And all we have to do is maintain the right balance so that this living organism lives as long as possible. There you go. And so in addition, all these vital elements, all these vital ferments, they play a role in the life of the earth. Because these vital ferments, well, they help to bring back organic matter. They clean up the earth. They clean all that up. And so they play a role in terrestrial life as they return organic matter to a mineral state. There you go. There you go. And the theory, I'm reaching the end of this theory, so I hope I've provided you with some insights here. MICROZYMAS are actually our ancestors. They work together to build us, to organize us, and life is particularly linked to organization. Without organization, there is no life. Upon the death of an organism, microzymas are released. They then act as autonomous ferments. Microzymas are the source of living matter, as we've seen. And living matter is only lent to us for a time. It continues its earthly life after us. Microzymas continue after us. And nothing falls prey to death. Everything falls prey to life. There you go. I have some experiments for you if you'd like. I'm going to simply read one or two experiments that Besham conducted because it's really interesting. And I'm going to read. So there's a gentleman, Mr. Donnet. Mr. Donnet had brought him an ostrich egg. Shaken. The goal was, I was talking about organization. We don't live, everything is organized, which allows us to live, meaning that each vital center, each thing, each environment has its role. And so the goal was to demonstrate that if we disorganized, the microzymas could no longer do their job. And so with a small egg, it was a bit difficult to shake an egg to the point of disorganizing it. It's so well organized inside that it's impossible to shake the egg without breaking the shell, to disorganize the egg. And so what he did was he tried with ostrich eggs. So they did this in 2 stages. The first time he brought him the egg, but he had opened it, so he wanted to see what happened with the closed egg. So he had opened it afterward, still. And so he was able to analyze what was happening inside. So it took time. It's like everything, That's it. The problem is that we want to have answers to everything all at once. People who delve into the subject, it takes time. That is to say, fermentation doesn't happen just like that. Et surtout, yes, these experiments are very long most of the time. It's precisely thanks to this patience that he managed to discover all this. Certainly the egg is organized, cleverly organized. And what precautions to ensure nothing naturally disturbs the admirable order that reigns there. Embryologists have admirably described all these parts. But after noting these wonderful arrangements, have they sought what is endowed with transformative activity in the egg? What is truly alive? What weaves the cells, the tissues of the egg that will emerge from it? And if they have sought it, have they recognized it? While waiting for me to answer these questions, let's ask ourselves what happens when we scramble everything in the egg? By vigorous shaking, it happens that what on the plan, in the divine plan, constituted a premeditated arrangement, something structured, built for a determined purpose, has been destroyed. So that the things that in the structure were meant to remain separate have been confused. Subsequently, the desired result is no longer achieved, even though the necessary material is still present. So what has really changed in the conditions? Few things in appearance, but the essential in reality, without which the matter will remain sterile. There, he really demonstrates that organization is important. It's that everything has found its place, and that organization is something that happens. It's a community of microzymas that act together. And matter, though inert and still present, is no longer able to live under those conditions or to form a living organism. Yet, what was just now capable of producing a chicken with its future, is it absolutely destroyed by shaking the egg? No doubt it's a dead egg, to speak like Mr. Donnet, but in the chemical sense, is it a corpse? No. Since an activity is manifesting, I say an activity is manifesting through putrefaction. From the moment an activity manifests, it means there is life. And Besham understood that well. And if you say that to scientists, no, well, no, for them, if there are molecules, chemical transformations and all that, if there is transformation, it means there is life. Another experiment. They have a reason to be. He buries a newborn kitten in chemically pure precipitated creosoted calcium carbonate. Creosoted, it's phenic acid, it's to prevent airborne germs from intervening. The whole thing placed in a glass jar, protected from dust, yet allowing for aeration. Aeration is important because oxygen also plays a role at some point in putrefaction, particularly in fermentation. After 7 years, the result is examined. The upper layers of calcium carbonate are intact. But where was the corpse? Things change in appearance. Of the kitten, only a few bone fragments remain. Everything else, even the fur, had disappeared. The calcium carbonate examined under a microscope looked like chalk. It moves me, except for the small aragonite crystals usually seen there. The microzymas were easily recognizable by their shape, their shiny appearance. It's a kind of artificial chalk. Thus, of the entire organism, only the presence of microzymas remained as evidence, just as the geological microzymas in sedimentary terrains are merely remnants. Of organisms in fruits, in eggs. I am very moved by all this work that is so little recognized, because we have the essence of life there, we have everything. And in the chalk, what he understood before precisely 1863, when he realized that ultimately it probably came from there, and that these microzymas were found in living organisms, he had discovered the microzymas in the chalk. In the chalk, there were small microzymas. This is probably the cause of Pasteur's theory. It's that protected from airborne germs, in his experiments shielded from airborne germs, he added chalk. Without. Imagining that there could be microzymas in the chalk. And so he had systematic reactions, and he said, ultimately, we haven't managed to obtain a sterile environment, so we can't say we are. There you go. And it's it wasn't clear. And so there you go, these microzymas from the chalk, he analyzed them, and you realize in the chalk they are probably completely preserved. That is to say, the microzymas found in the chalk are the trace of living organisms that date back a very, very long time, very, very long time probably. Because the microzymas, and it's Hector Grasset who explains this well, they are the principle of the basis of putrefaction, so they do not putrefy. They can be killed. That doesn't mean disappear, but killed. By temperature, above 100 degrees, if kept at 100 degrees for a long time, they are killed. That is to say, they are still there. They can't be destroyed like that. They are still there. But if extracted, they can no longer ferment. They can no longer produce enzymes. No, I should say in reverse order, produce enzymes, but rise. Nor multiply, even more so. Antoine Bechamp, the microzymas from the chalk, he extracted them. He managed to make them multiply. And we think he managed to revive microzymas from a distant time. They are, however, they are not protected by the chalk, by the calcium carbonate. They can still be destroyed by the elements. Since Bechamp managed to analyze their composition, he indeed managed to destroy them. but by force, by the force of the elements, not naturally, not by putrefaction. As long as we don't intervene, they don't move. And inert matter, if we don't intervene, doesn't move. And if a leaf, a fruit, in an animal decomposes, it's thanks to the ferment of life, which carries out this decomposition. It's huge, you know. I don't know if you realize how it completely changes things. That's why. It's not obvious, and it's not easy for me either to keep repeating all the time. And I'm afraid that when I've already expressed all this, I'm afraid of sometimes losing track. But yeah, I think it's a completely different perspective. And so from the perspective of viticulture, naturally, you're going to have questions. I don't know if you've already sensed the importance of the boundary between the environment and the fact that the ferment is constantly nourished. So in the stages, in the experiments Bechamp conducted on wine fermentation, even though he talked about ferments as being air ferments, all these experiments are really very, very interesting because they provide a number of elements that might escape you because I don't know what you know compared to the little I know, and if there are things that have been overlooked. For the people who continued in winemaking afterward, so he is well aware that the care given to cultivation is important. The respect for the vine is very, very important. He talks about it, but he doesn't elaborate on the subject. He hasn't analyzed things. He made some observations, but he hasn't analyzed things. And undoubtedly, yes, biodynamics brings elements. There was someone else who was supposed to give me some insights on biodynamics, but who is currently studying everything related to health. Is it the 500? Can we call it that? There you go, the manure. He told me he was analyzing all this from a perspective, from Bechamp's point of view. He sent me an email this morning saying, I'm sending you a few things, but I haven't finished yet. So I forwarded that to Frederick, but I think he might have very interesting things to say. So he observed, he himself said that a field on which we spread, the manure thus energized and diluted already said that the consistency was different. It felt like walking on moss. So I suppose, and you'll see that with Frederick, and I'll check with this person who wants to share all this. Et donc, you'll probably learn elements from that perspective. So what I've seen is more from the grape, the production of the grape. So the challenge for the winemaker is to produce quality wine, but more specifically to prevent the wine from turning. I think that's the crucial point. A wine matures over time until the moment the fermentation shifts. The key is to find the right balance. I say we're on the razor's edge, I think. Especially since I explained to you the microzymas adapt, meaning the environment has transformed during winemaking. And at some point, the microzymas might take time to adapt. It might not happen overnight. So we have an adaptation phase, but at some point they could start feeding directly on alcohol. And the transformation, and the wine is the wine turned. But the quality of the wine, because one might think it would be enough to get rid of the microzymas. So filtration removes microbes, but not microzymas. Because filtration doesn't reach particles of nanometric size, smaller than the micro in any case. Et donc, but is it wise to get rid of them? Because what is it that makes a wine high quality? But I believe the quality of the wine is precisely due to the fact that it's alive. So yes, in terms of taste, it's subjective. The contribution of quality is completely subjective. But if we analyze it differently, theoretically, a quality wine should be able to, from a health perspective, observe that its properties in terms of bioelectronics are compatible with health. On one hand, and that's the case with good wines. From a MICROZYMA perspective, well, I think there was a time when we heated the wine to prevent fermentation, to stop the wine from spoiling. Yes, I believe a good wine should allow us to observe these microzymas, see if they're still alive using a dark field microscope. And then to go further in the experiment, see if they are. But it's not complicated to recover a bit of ferment. It's nothing. I think it is. I visited people who made organic wines from Claret de Die. They explained that they bottle very quickly and actually expel the ferment by uncorking quickly. It's a jet and the cork pops out. He told me, but anyway, the ferment is dead there. And I don't think so. I don't think the ferment is dead. I think the ferment is still alive. So see if it's still alive. And I think that if we want a good living wine, you have to trust your instincts, your experience, and maybe try things out. So I add something that seems logical to me. That's my logic. I am completely anti-conformist. A little conformist for me, when everything is framed and when we see how science has followed a path and all scientists follow the same path, I suppose it's the same for wine. It's just that you've all probably followed techniques that were taught to us, and maybe there are 1,000 other ways to look at things. And I was thinking, for example, yes, I really recommend Bechamp's book on wine fermentation because it shows the effects of different things. The balance between water and sugar, especially in the must. The action of oxygen, a whole bunch of things that will be interesting to see. And you have the experience to analyze that, to take what might be interesting for you. But I was thinking especially about sugar. Photosynthesis is done by the green parts of plants. And I wonder, by pruning the vine as we do, is it necessarily a good thing? Isn't there another way to do it? That's what inspired me too. In some countries, I don't remember where I saw it. Was it in Spain? I don't remember. They treat the vine as a creeper. Yes, they do it in. Yes, they train the vine to climb. In fact, they cultivate differently. And I think these green parts, if we prune them a lot, we'll definitely have less sugar. Well, I think so. We might have fewer bunches too, so I don't know. Maybe the sugar's more concentrated. I don't know at all. I don't have any data. I just think maybe we should be wary of everything we learn and the techniques that are a bit formatted, and maybe look elsewhere and then practice our art, because it's obvious it's an art. It's an art, and so experience will pave the way. There, I've reached the end. So the lessons on wine fermentation, I'll send you the link then on this page of my website. My site is called Bonnish Habitudes Afar. Antoine Bechamp's theory, you'll find everything related to Antoine Bechamp's theory. What hinders the temperature? Is that all? From what Bechamp analyzed, he managed to kill them with temperature. He subjected them to extreme chemical treatments. They resist. They can sometimes be in situations where they can't express themselves, so they become dormant. They will be dormant, but that doesn't mean they're dead. They might be in an environment where they serve no purpose, so they just are. When we preserve them in chalk for centuries, there's something. It extracts them, it makes them multiply anyway. It's pretty incredible. And during all that time, they remain inanimate. So there you go. However, yes, they can be in an environment where they won't express themselves. But if they don't express themselves, they have no interest in those cases either, because I think it's when they express themselves. Although in wine, if you want a proper balance, and that's precisely the balance of wine, it's reaching a stage where there will be such a balance that the microzymas will no longer ferment if you want the wine to remain stable. But actually, wanting stable wine is going against nature. Because everything is nature, it changes constantly. What's amazing is that microzymas manage to maintain a consistently stable environment in living organisms. Stable by evacuating and accepting or evacuating just what is needed in order to always keep the environment stable. And it's extraordinary. That is this homeostasis they manage to maintain in living organisms. It's almost magical. It's fabulous. Everything emits vibrations. So what precisely distinguishes living matter from inert matter is the microzymas. And you can't have deep chemical transformation in a stone. You won't have fermentation, this kind of thing to break them, you know. So that still comes back.
Il y a quelqu'un qui a...
Someone won a Nobel Prize in 1907. It's Edward Buchner. Precisely for enzymes. It's enzymologists don't know Bechamp, but they know Edward Buchner. Edward Buchner won this Nobel Prize because he showed that it was possible to carry out fermentation with the enzyme alone. And when I saw that, because I discovered all this not very long ago, actually, I thought to myself, something's wrong, it's not possible. Antoine Bechamp explains that the enzyme only intervenes in the first phase. The enzyme alone, if we remove the ferment. In sterile conditions, you know, aseptic, if we only have sugar and water and the enzyme, only the enzyme, which is soluble, it's the enzyme. And with only the enzyme, we will never achieve fermentation. We will never have alcohol. And Bechamp shows that, and I wonder how Edward Buchner could have won a Nobel Prize. And at one point on Wikipedia, it talked about Edward Buchner, and then it said, here, he won his Nobel Prize because he showed that the enzyme could carry out fermentation. Thanks to the addition of a rock of some kind, I don't remember, of some kind of rock.
Ah, vas-y, mais attends.
Wait, there's not just the enzyme in the solution. There's not just sugar, water, and the enzyme. There's a sedimentary rock. He adds a sedimentary rock in there. And what's in sedimentary rocks, if not ferments, if not microzymas? Living beings that were in there. There you go. And actually what causes the fermentation is the rock. It's kieselguhr, I'm not sure what. Yeah, that's it. And what causes the fermentation is it's the ferments that are in the rock. If he doesn't add this rock, he doesn't have fermentation. And this has been repeated 8 times, and he won a Nobel Prize for proving that enzymes were capable of fermentation. It's to judge a little bit the level of science. But it's huge. That's the question that arises. We don't know yet. It's the but, and especially when we talk about DNA, it's important to know, and this too, it's part of the things that by peeling back science, by making the connection with contemporary science. It's important to know that microzymas are at the origin of DNA. I was saying that they are at the origin of everything in our organisms, but particularly of DNA. When we take the DNA of a new individual, it is different from that of its parents. He inherits it, but it is different from that of his parents. And there is a publication, there are the rare publications we can find from researchers who try to continue Bechamp's work, scientists who have shown that DNA was self-assembled thanks to granules inside the yolks. So egg yolk. And so DNA itself is produced by microzymas, and undoubtedly all the microbial genes we have in our bodies are produced by microzymas as well.